Okay, so let's just do one demo for Blast and Psi-blast. So in this case, you just need to input one sequence. So here you can see this format is called a FASTA format. A FASTA format start with a greater sign. It doesn't have to be two, just one is good enough. Some gene name, some description, and sometimes people put a species name. So this is actually rice. And then these are the amino acids for the sequence. So it's called so-called FASTA format. Okay, so here shows this the blast. So you just go to this portal. It actually has all kind of blast. This is actually for nucleotide sequences. Then you can do other types. But for protein, you go to protein blast. It's actually very easy to use. It's also very efficient. You can get results in real time. So all you need to do is just copy and paste the sequence here. Then you can actually specify, for example, you can find a database. So typically you use this so-called NR, that's a so-called non-redundant protein sequence. It is huge. It's a collection of pretty much all the major protein sequence databases. And then just remove the redundancy. The redundancy bar is very low. That means if two sequences have a one amino acid difference, it is actually treated as two entries. So only those 100% identical proteins, they remove the one of them. And then you can use this so-called ref-seq. You actually, which is a little bit, the less redundancy. And then you can use model algorithm. You can use a UniPro. You can use this structured database, etc. So let's say just find this UniPro. And then you can just do a direct blast. You can do Psi-blast. You can do some other pattern blast. And then you just hit a search. So in this case, we just want to see, let's say, UniPro hits. Yeah, this typically takes anywhere from maybe 30 seconds. Okay, so yeah. So it doesn't have a significant hit. Then you can use NR to search. So if it doesn't have a, let's say, UniPro hit, then you can use NR to search it. So that's why you see that UniPro, even though it is high quality, but it is not that comprehensive. I mean, a lot of proteins are not covered by UniPro. Nowadays, this is just sequences are so many, and then you may not be able to find that protein in UniPro. So that's why you need to use other tools. So here, this actually is a gene in rice. So actually one of the investigators at Life Science Center, he gave this to me and I did some study. So you see that in case they're not in the major databases, then you can actually just see what kind of information you can get. And then you can see some hits. Many of them are so-called hypothetical proteins, uncharacterized proteins. So that means you don't know the function. But then you start to really see whether any of them contain some functional information. So in this case, none of them have had any function annotation. So you don't know what this protein does, right? But then you can see some information. For example, one thing you can see is tree structure. You see that what kind of species have this gene. Interestingly, it's pretty much in plants, and mostly in the so-called monocots. And those actually is one special type of plants. So you know this gene is relatively so-called young gene. It was not conserved in evolution, but rather in evolution is a kind of so-called emergent new gene. So that gives you some idea. And then you can also see other information. Let's say it's a multiple sequence alignment. So we'll talk about multiple sequence alignment, but here it is not the true multiple sequence alignment. That is this alignment is based on just one sequence against others, instead of treating all the sequences equally. Because when you treat all sequences equally, computation time will be quite a lot. But here, this is a so-called heuristic search. Then you see, what kind of sequences you get, and then which one is conserved. So here you start to see the conservation patterns. So there are many. And then start from here, you can do phylogenetic tree analysis, which we'll talk about that later. So here, that's just one direct search. But then you can also do this so-called Psi-blast search. That means that you do this iteratively. Then you see whether you can get more information. So the message I want to convey here that majority of cases, you can simply go to major databases like UniPro. You'll be able to find a lot of useful information. But if it's a new gene or some special function or some interesting feature you'd like to explore, it may not be included in those portals. So that's where you need to go deep using different tools and then get some new information from those tools and those databases. So it, of course, requires you to know more features of the tools and also more tools. So here, this is the way you get this Psi-blast. And then it basically generate one round of hits from a sequence database. And then Psi-blast internally uses sequences to build this profile. That means where is conserved, where it's not conserved. So this is all based on query. We call it star alignment. That means that all the other sequences will align to the star. The star is the query protein. And then you see the profile. So this profile actually is not really true multiple sequence alignment of all these proteins, because it will be just too time consuming. So there is some catch, but still it is pretty useful. So once you really run this Psi-blast, then you can see some new hits. And then that new hits actually may provide some functional information or some other useful information. So that's kind of the idea behind it. And then as you can see in the previous page, you actually can select and deselect what sequences to include to build a profile. So these are a lot of features you can explore. So all these bioinformatics tools, I mean, even as simple as the blast, there are just so many features. I even saw a whole book about the blast. So there are just so many ways to do it. Of course, if you are so-called power users, that means that you are better than most others, then you really can do the command line based blast search. You can even run blast locally on the machine, which may give you capacity to do batch analysis. That means that instead of running one protein at a time, you can handle many. So here you see that it starts to give you some new proteins. So here, this highlights new proteins identified. And then you start to see some hints about function. For example, this chloroplast-like protein, this CHUP1, it has some functional annotation. Compared to all others, pretty much hypothetical. So this is one way you can find some functional information through this Psi-blast if you don't know anything about this protein. Yeah, majority cases, you'll be able to find useful hints about this protein. Of course, there is a catch. That is, some proteins, they are so-called orphan genes. That means that people cannot find any significant similar proteins to that gene. Then you won't be able to do this approach. Of course, we will have other ways to handle this kind of so-called orphan genes. But at least the Psi-blast is a powerful tool to find related proteins. And then from that related proteins, if you know the structure, function, or other information, then you can transfer that information as the inference to your query protein. Okay, so that's the end of a demo of a Psi-blast.